I did some of the research, okay, so I can show you some slides. And this was some data I presented at 2 Iridoid symposium meetings ago. Okay, so in 2010. So some DNA detection activity by the 2 major Iridoids in DAA and AA. And we use DAA and AA again because their names are too long to say. And we're lazy, we don't want to have to say the words all the time. So DAA and AA. Now, we had done an experiment in our labs that you hopefully got to see, where we took some cells, some individual cells, and we exposed them to this chemical here, this one here, that I'm pointing to here. It has, again, a long name, but we call it 4NQO for short. But this chemical, 100% of the time, will damage DNA in the cells. This, when you do research to see if a chemical has any cancer activity, you will test that chemical in cells, similar to the test we've done here, but to make sure those cells are normal cells and they respond normally and they're not some kind of super mutant cell that's resistant to any chemicals, you use this 4NQO to test to make sure that those cells are normal and that when you expose the cells to this chemical and DNA is damaged, then you know, okay, we can move forward and use these cells. They represent the normal cells in our body. So it will damage DNA 100% of the time. And that's shown here. What it does is it causes breaks in the DNA. And when the breaks in the DNA occur, that leads to a lot of health problems. Okay. Enough breaks occur, the cell has to repair those breaks. If the breaks are not repaired appropriately, we can have other health conditions occur. That would include cancer. Okay. Now, we repeated this experiment again with the same chemical, 4NQO. But we added the iridoids from Noni individually, doing separate experiments, and then measured the same amount of DNA damage that would occur. So we again are measuring DNA damage. When we added the iridoids from Noni, that DNA damage was abolished. It protected, there was no breaks. We could not measure any significant increase in DNA damage. So graphically, this is what it looks like. So this is there's a term that we use called genotoxicity. Geno meaning, DNA or genes. They're toxic to the DNA. The blank means the amount of DNA damage that occurs spontaneously. So you'll see, if I can get this to work again. Right here. So this is the blank, right here. This is just the cells with no exposure to 4NQO. So very low level of spontaneous DNA damage that occurs. That's normal. There's no increased risk there. When we add the 4NQO, you see the big red bar. That 6-fold increase means 600%. So here it says percent increase. You need to add it times by 100. That's 600% increase in the amount that we add to this 4NQO. Now, when we have the same amount of 4NQO, and then we use DAA, or the same amount of 4NQO plus the AA, you'll see there in those 2 little areas to the right here, it was back down to the same amount as the blank, back down to baseline level. So what this said here is that in this test, the DAA and the AA absolutely protected these cells against the DNA damaging effect of that chemical. Okay? So this is a big thing. We actually published that, as you see down below there. It's published in a peer-reviewed journal called Food Research International. And we included some other information on the iridoids in Noni fruit. But this was a big finding. Now, why do we care? Well, one, it's in Noni, so we care. We don't know what Noni does. But we needed to explain something. You see, we had funded a clinical trial at the University of Illinois College of Medicine that was conducted by Dr. Mia-Ning Wong, a member of the pathology department there. This study looked at the effect of original, or now original, the original bioactive, okay, product, the effect that that had in heavy cigarette smokers. So these people were smoking 19 or more cigarettes a day. That's a pack of cigarettes a day. Okay? There's a reason why smoking is associated with cancer, because the chemicals in the cigarette smoke break the DNA. They damage the DNA. Okay? And that's what was measured here, DNA damage. So we measured the amount of, you see in the bar on the left side, the amount of DNA damage that was occurring in the white blood cells of these smokers. The reason we took the white blood cells out is because we can't take, liver tissue or lung tissue. You can draw white blood cells out and measure DNA damage easier than that. People will agree to that study a lot more easily than if you remove something else from them. Sorry, that's a scientist joke. It probably doesn't translate. Okay, but basically, after 1 month of drinking original, there was a 44.9% decrease in the amount of DNA damage that occurred. So we knew there was something special about noni. We knew there was something special about noni compared to the placebo used in the study, which had grape juice in it, which is a good, healthy fruit juice, right? There's antioxidant activity, but there was something unique about noni. Again, what was unique about noni is the iridoids that protect the DNA. Okay, now not to go too long, and leave enough time for alpha, I'm going to share some antimicrobial activity of the iridoids in noni. Now in this experiment, Shishing isolated the iridoids in an extract from our noni fruit that contained DAA, AA, and scandocyte. And we incubated pathogenic microorganisms in the presence of these iridoids. So what that means is we took these iridoids and we gave them to bugs that cause food poisoning, okay, or some other disease state. And we wanted to see if the iridoids would kill them, basically, or keep them from growing. Here's what we see for E. coli. Now, as the graph goes down, that means there's less E. coli cells that could live or could grow, okay? And as you go along from left to right, that's the increasing concentration of the iridoids in noni, okay? So what this shows is that with more iridoids, you had less ability of the E. coli to thrive and grow, okay? This is a good thing. This is a good thing. This is also one of the reasons why we don't need to use preservatives in our beverages. We don't need to put in chemical preservatives because the stuff in there kills them. Kills the bad stuff, okay? Okay, Candida albicans. This is a problem. This is a yeast that causes a lot of problems for a lot of people. This organism was particularly sensitive to the iridoids. We saw, in fact, even greater inhibition of the growth of Candida with the iridoids. And also Staph aureus, heard of staph infections or other things like that. Anyhow, they also were sensitive to the effects of the iridoids from noni. So here's a couple examples: antimicrobial, antibiotic type activity, and DNA protective activity. So now in extract, we bring in a new set of iridoids.